For 70 years, we've treated biology like a chemical hard drive. You look at DNA and you see a sequence, a long, linear string of code dictating how an organism is built, one molecular bit at a time. But treating biology strictly as a sequence leaves some massive gaps. If life is purely a localized chain of chemical reactions, it becomes incredibly difficult to explain how millions of cells instantly synchronize across an entire organism to maintain coherence. That gap in understanding has opened the door to a radically different framework. Instead of looking at genetic data as a static chemical code, a new school of thought treats it as a nested informational structure, a system governed by phase topology rather than just physical bonds. You are looking at two entirely different ways to interpret reality. You either assume biological complexity is the result of countless chemical accidents accumulating over billions of years, or you view it as an active computational optimization process. How you answer that dilemma dictates exactly which tools and theories you will use to interact with life itself. This debate is pulling theoretical physics straight into biology. We are now seeing radical concepts like the A model and infodynamics encroaching on the discussion of how living systems organize themselves. And these aren't fringe ideas. This published article explicitly asks if physical forces like gravity are evidence of a computational universe. These theories are being seriously proposed in the realm of classical physics. The core thesis of these new models is that biology operates as a computational matrix. In this view, living structures are managed by phase coherence, waves of information synchronizing across a field, not just molecules bumping into each other. For a pragmatic empiricist, that is a tough pill to swallow. Relying on elegant, system-wide theories means walking away from the hard, localized certainty of proven chemistry. So let's look at the actual trade-offs. We are going to evaluate the standard biochemical sequence against the nested phase structure across three distinct metrics, separating the hype from viable scientific theory. This matrix outlines our evaluation. We compare the standard biochemical model against the nested informational model. First row, the storage medium of biological data. The standard approach treats data storage as strictly localized. A gene is a physical sequence of nucleotides at a specific address. This gives us precise targeting tools like CRISPR. You can find the sequence and cut it. But it treats the actual 3D geometry of the DNA as secondary to the letters themselves. The nested model offers an alternative concept known as long-range phase memory. Instead of reading letters on a string, this model treats the physical helix of the DNA as a phase waveguide. It suggests the structure distributes phase information across a topological lattice. Because a waveguide maintains a consistent phase state across its entire length, distant regions of the molecule can react as a single unit. This mechanism provides a formal explanation for how a whole organism maintains synchronization across its entire system. The standard model wins on precise physical manipulation. The nested model provides far superior explanatory power for systemic coherence. Let's move down to the second metric on our board, structural stability, specifically how we understand complex protein folding. The standard model predicts the final shape of a protein purely through localized bonds and chemical thermodynamics. The sequence dictates the folds, but simulating those folds atom by atom requires massive brute force computational power. And even when you get the right shape, the simulation rarely tells you why the system preferred that specific geometry. The nested model sidesteps the atom-by-atom approach. It treats proteins as composite knots, viewing their shapes as geometric solutions to informational constraints. This plot maps a stable, coherent configuration in a mathematical field. Under this model, we view a folded protein as a physical realization of this geometry, a structure fixed in space by its own topological requirements. The cost of using this theory is steep. To map those geometric solutions, you have to trade standard biochemistry for highly abstract, non-commutative algebra. Treating biological structures as topological knots elegantly solves the folding puzzle conceptually, but remains difficult to translate into practical engineering. That brings us to the final and most foundational metric on the board, the primary driving force of the universe. The standard model anchors itself to the second law of thermodynamics. It dictates that isolated physical systems universally tend toward disorder and entropy over time. But biology presents a glaring contradiction to that rule. Pure thermodynamics struggles to explain why living systems consistently generate highly symmetrical, complexly ordered structures. The theoretical counter to this is the second law of information dynamics. This law measures information entropy. It suggests that high symmetry and even gravitational attraction are actually data compression mechanisms, operating to reduce the total computational power required by the universe. If physical laws are actively minimizing processing load through compression, then reality functions as an environment governed by its own source code. Accepting infodynamics means accepting that the universe behaves like a system optimized for computational efficiency. Thermodynamics perfectly governs the chemistry and the hardware of the universe. Infodynamics claims to govern the information optimization, or the software. Deciding between these frameworks isn't about declaring a universal winner. It comes down to the specific problem you are trying to solve. Look at this decision tree. We can split the paths here based on whether you want to manipulate local biology or understand systemic design. Path A is for the pragmatic empiricist. This is the route for medical researchers, gene editors, and bioengineers. If you need to cut a gene to cure a localized disease, stick to the standard sequence model. It produces actionable wet lab results. You simply have to accept macro-level coherence as emergent noise that you cannot yet control. Path B is for the systems thinker. This includes computational biologists, theoretical physicists, and AI developers. If your goal is to map how a whole system synchronizes, invest your time in topological field theory, phase manifolds, and infodynamics. Just prepare to navigate a massive amount of theoretical noise before you hit practical application. The universe might just be a chaotic biochemical soup, or it might be a nested quantum neural network. But the ultimate value of either framework isn't determined by its elegance. It is determined by the utility it brings to your specific work.